Microalgae Biotechnology for functional ingredients and Omega-3 production. Advances, health benefits, and commercial perspectives. Microalgae biotechnology has emerged as a versatile and rapidly advancing platform for the production of high-value functional ingredients, nutraceuticals, and sustainable omega-3 lipids. A wide diversity of microalgal species has been explored for their biochemical richness, offering proteins, pigments, polysaccharides, and polyunsaturated fatty acids, PUFAs, with significant health-promoting properties and commercial relevance. Among the most extensively utilized organisms is Spirulina platensis, a cyanobacterium traditionally consumed as food and now widely marketed as a dietary supplement. This organism contains exceptionally high protein levels, reaching up to 70% of its dry biomass, with a complete profile of essential amino acids and high bioavailability. Its carbohydrate fraction, approximately 15%, consists mainly of polysaccharides, while lipids account for approximately 5%. A defining feature of S. platensis is its content of phycobiliproteins, particularly C-phycocyanin and allophycocyanin, which serve as light-harvesting pigments. These compounds are valuable as natural food colorants, cosmetic ingredients, and fluorescent probes in analytical applications. Importantly, phycobiliproteins exhibit antioxidant, anti-inflammatory, and hepatoprotective activities largely attributed to their tetrapyrrolic chromophores, phycobilins. Their potential roles in photodynamic therapy and cancer treatment further highlight their biomedical significance. Another commercially important species is Dunaliella salina, recognized as one of the richest natural sources of beta-carotene. Under stress conditions such as high salinity and light intensity, this microalga can accumulate beta-carotene up to approximately 14% of its dry weight. This carotenoid is widely used in food, pharmaceutical, and cosmetic industries due to its provitamin A activity and antioxidant properties. Industrial-scale production has focused on optimizing cultivation parameters and employing efficient extraction methods, notably supercritical CO2 extraction, which is well suited for recovering nonpolar compounds like beta-carotene. Species of Chlorella, particularly Chlorella vulgaris, are also prominent due to their rich composition of bioactive molecules. These include proteins, vitamins, and pigments, alongside demonstrated biological activities such as antioxidant, antibacterial, hepatoprotective, and immunostimulatory effects. Experimental studies have also suggested potential anti-tumor properties, making Chlorella a valuable candidate for functional food and nutraceutical applications. The unicellular green alga Haematococcus pluvialis is notable for its ability to synthesize astaxanthin, a high-value carotenoid with potent antioxidant activity. While it produces primary carotenoids under normal growth, stress conditions trigger the accumulation of astaxanthin up to 2 to 3% of dry biomass. This compound is widely used in aquaculture, dietary supplements, and cosmetics due to its role in reducing oxidative stress and supporting eye and skin health. Red microalgae, such as Porphyridium cruentum, represent another important group, producing sulfated exopolysaccharides, phycobiliproteins, and PUFAs. Although lipid content is relatively low, approximately 6.5%, these lipids are enriched in biologically important fatty acids such as eicosapentaenoic acid, EPA, arachidonic acid, and linoleic acid. These PUFAs are associated with reduced cardiovascular risk, anti-inflammatory effects, and potential anti-cancer activity. The sulfated polysaccharides also exhibit antiviral and immunomodulatory properties, enhancing their functional value. Microalgae are particularly significant as primary producers of long-chain omega-3 fatty acids, including EPA and docosahexaenoic acid, DHA. Species such as Phaeodactylum tricornutum can contain 30 to 45% PUFAs, with EPA contributing up to 40% of total fatty acids, while Monodus subterraneus also demonstrates high EPA yields. Additionally, Schizochytrium is widely used for industrial DHA production. These omega-3 lipids are essential for cardiovascular, neurological, and visual health, and have been associated with reduced risk of coronary heart disease and improved cognitive development. From a bioprocessing perspective, the selection of microalgal strains requires detailed compositional screening to identify target metabolites such as antioxidants, pigments, polysaccharides, and PUFAs. Subsequent optimization of cultivation conditions, including light intensity, salinity, nutrient availability, and temperature, is critical to maximize productivity. Microalgae exhibit metabolic flexibility, growing under photoautotrophic, heterotrophic, or mixotrophic conditions. While photoautotrophic cultivation dominates large-scale production, mixotrophic and heterotrophic modes often yield higher biomass and productivities, albeit with increased complexity and contamination risks. Cultivation systems are broadly divided into open ponds and closed photobioreactors, PBRs. Open raceway ponds offer low capital and operational costs, but suffer from contamination risks, environmental variability, and limited process control. In contrast, closed PBRs provide controlled conditions for light, temperature, and nutrient delivery, resulting in higher biomass yields and reproducibility, though at greater capital investment. Contamination remains a major challenge, particularly in large-scale systems necessitating selective growth conditions or closed cultivation strategies. Downstream processing represents a significant cost component, often accounting for 20 to 30% of total production costs. Due to small cell size and low biomass concentrations, harvesting requires energy-intensive steps such as flocculation, centrifugation, filtration, or flotation. Efficient extraction of intracellular products typically involves cell disruption followed by solvent extraction, with increasing emphasis on green technologies such as supercritical CO2 and ethanol-based processes to ensure sustainability and product quality. The commercial landscape for microalgae-derived omega-3 ingredients is expanding rapidly. The global market, valued at approximately $1.19 billion in 2024, is projected to reach around $1.81 billion by 2030, driven by a compound annual growth rate of about 7.2%. Growth is fueled by increasing demand for plant-based, sustainable, and clean-label nutritional products, alongside heightened awareness of the health benefits of omega-3 fatty acids. Microalgae offer a direct and sustainable alternative to fish oil, as they are the original producers of these fatty acids within marine ecosystems. Major industry players, including ADM, DSM, and Corbion, are actively investing in large-scale production technologies, strategic partnerships, and acquisitions to strengthen their market position. Industrial production increasingly relies on controlled fermentation systems, where microalgae are cultivated using renewable plant-based feedstocks, such as sugars, enabling scalable, low-impact manufacturing with reduced land, water, and carbon footprints. In summary, Microalgae Biotechnology represents a highly promising and sustainable platform for the production of functional ingredients, particularly omega-3 fatty acids. Continued advancements in strain selection, cultivation technologies, and downstream processing are expected to enhance economic viability and support the growing global demand for health-promoting, environmentally sustainable products.